So hi, I'm Shiyu. I'm a research fellow at the Psychosis Neurobiology Lab at McLean Hospital. And today I'll be presenting on Thalamo-Cerebellar and Thalamo-Cortical functional connectivity in the early stage of psychosis. Oops. So this presentation will present evidence that thalamic connectivity changes dynamically during the early stage of psychosis and will cover three main sections. The first looks at contrast between patients and controls. The second looks at a qualitative assessment of trajectories over this period. And the third looks at quantitative models of the trajectories. So what is the early stage of psychosis? So this diagram is a putative model showing the different stages of the lifetime course of psychosis. It starts with a preclinical phase, followed by a clinical high-risk prodromal stage with attenuated symptoms that leads to the onset of the first episode of psychosis, which usually occurs in the late teens or early 20s. This is followed by the early stage of psychosis, marked by repeated episodes of psychosis of variable duration and variable inter-episode remission, and finally a more stable chronic phase. So in this study, we define the early stage of psychosis as the first 5 years after the onset of psychosis. We are interested in this period because it's a dynamic critical period with heterogeneous symptom presentation and outcomes. So for example, recovery could occur after the first episode or after repeated episodes. It has been proposed that the right intervention could result in disproportionately favorable outcomes. So why is thalamic connectivity important? The thalamus has been shown to have extensive connections with both the cortex and cerebellum, and is a key area for information processing. It acts as a relay for motor and sensory information, so from the peripheral nervous system to the cortex, as well as an integrative hub for cortical information, thus integrating information across multiple functional networks. So the thalamus is involved in important functions, such as voluntary movement, sensory gating, or the filtering of sensory information, and cognitive flexibility, which is the ability to switch between different concepts. Previous research has shown that thalamic connectivity is abnormal in both the early stage and chronic stage of psychosis. So compared to healthy controls, patients have been observed to have hyperconnectivity with motor and sensory cortices, hypoconnectivity with the prefrontal cortex, as well as the cerebellum. And in clinical high-risk populations, cerebello-thalamo-cortical hyperconnectivity has been shown to be predictive of time to conversion to psychosis. And finally, in abnormal thalamic connectivity has also been linked to psychosis symptoms and cognitive dysfunction. However, most of these studies were primarily cross-sectional, and none of them have explored longitudinal dynamic changes in thalamic connectivity. These studies also mainly looked at anatomical regions, so the brain is parcellated by anatomical regions, and few have looked at thalamic connectivity with respect to resting state functional networks. So this study addressed three questions. Is thalamic connectivity with resting state networks abnormal or similar in early stage psychosis patients compared to healthy controls? Secondly, there's thalamic connectivity with different functional resting state networks through different trajectories over the early stage of psychosis. So whether or not trajectories remain relatively stable over time or whether they increase or decrease over time. And if the trajectories do change over time, what is the shape of the trajectory? Would it be mainly a linear trajectory or a curvilinear trajectory? And finally, there's the duration of illness, which we define as time since age of onset of psychosis. Is this a significant predictor of thalamic connectivity? So this is the design of the study. So 156 patients in the early stage of psychosis were recruited along with healthy controls. And out of these patients, we collected 233 scans. So about 77 scans were follow-up scans. These 233 scans were then divided into time points based on time since age of onset in years. So this means that at time point 0, participant scan data was collected the same year as their onset of psychosis. And at time point 2, for example, participant scan data were collected 2 years after the onset of psychosis. So the first objective of the study was to compare thalamic connectivity between patients and controls at their earlier scan data. And we do this by looking at general linear models. The second is to look at the shape of the trajectory in patients over the early stage of psychosis. And to model this, we use generalized additive models, or GAMs for short. And we use GAMs because they are flexible. They're not restricted to having a linear trajectory, but they can also model curved trajectories. However, this is only a qualitative assessment of a shape, as GAMs have the assumption that all the scan data will be independent, whereas we have longitudinal data which could be correlated. So to actually model thalamic connectivity trajectories quantitatively, we use linear mixed effects models. So these are the study methods. So patients undergo a 3T resting state fMRI scan, and the scan data is pre-processed in the contour box. First level analysis is a ROI to ROI bivariate correlation that is Fisher transformed. And the functional network ROIs are the 7 cortical ROIs postulated by Thomas Yeo and the 7 corresponding cerebellar ROIs postulated by Randy Buckler. So for this analysis, we excluded the cerebellar visual network as it has been shown to be poorly represented in the cerebellum. So this gives us a total of 13 unique thalamus to functional network connections. So second level group analysis, contrasting patients and controls were also conducted in the Quant toolbox. And within patients alone, we also looked at the qualitative assessment of their trajectories using exploratory GAMs. And final models were linear mixed effects models. So baseline demographics were similar between patients and controls, except for 2 variables, the scan age and the invalid scan count, which are the scan volumes that were excluded during pre-processing. So these variables were controlled for a later analysis. Another variable that was significantly different between patients and controls was MCAS total scores, which represents community functioning. And this is not surprising as patients have been shown to have reduced functioning compared to controls. Finally, our sample consists of patients diagnosed with affective psychosis as well as patients diagnosed with non-affective psychosis. And this sample had about 60% of patients diagnosed with affective psychosis. Yeah. So for the first objective, thalamic connectivity with functional networks were indeed abnormal in patients in the early stage of psychosis, even after controlling for scan age and invalid scans, and even after multiple comparison corrections using the false discovery rate. So similar to literature, on the left, we observe hyperconnectivity with the motor and sensory networks. And on top of that, we also observe hyperconnectivity with the dorsal attention network and the limbic networks. We also observe hypoconnectivity with the cerebellum. And because we use a cerebellum postulation, we find that this is specific to cerebellar regions linked to the default mode network and the frontal parietal network. Thus, we observe that thalamic connectivity abnormalities differ depending on which resting state network we're looking at and are not actually uniform across the brain. So for the second objective, we next looked at the shape of the trajectory over the early stage of psychosis. And we found that networks related to higher order functions, such as the DMN and the FPN, show dynamic changes over the first 5 years after psychosis onset. So these figures here show a comparison between the GAMs and the linear model. And in the case here of the Thalamo-Cerebellar FPN functional connectivity, the GAM is not significantly better than the linear model. And so we conclude that this connection follows a linear trajectory. For the other 3 networks, the GAM is significantly better at than the linear model at modeling the data. And this shows that these networks follow a curved trajectory. And what this means is that we add a quadratic term to our final mixed effects models. As our data set is semi-longitudinal, we have to take both cross-sectional and longitudinal effects into account. And we do this by separating our time variable, which is time since age of onset of psychosis, into an average variable and a deviation variable. So the average variable would be the same as the actual time variable if the participant only has 1 time point. But if participant has data at 3 different time points, so for example, if participant has data at time point 0, 1, and 2, then the average time would be 1. And so this represents cross-sectional effects. The deviation variable is the average time variable minus the actual time variable. And this term would be 0 if the participant only has data at 1 time point. And so this represents longitudinal effects. So we test if average and deviation variables have similar effects. And for all of our models, they were not significantly different from each other. And so we conclude that the cross-sectional and longitudinal effects were similar, and we combine them into the single time variable. And so for our third objective, quantitative modeling does show that time is indeed a significant predictor of thalamic connectivity, especially in networks related to higher order functions such as the DMN and FPN. And this is an example of one of our equations of our models. So functional connectivity is the outcome, which will be the thalamus to network functional connectivity. And we have 2 time terms. And the quadratic time term is only added if our trajectory is a curved trajectory. And for our models, we control for age, outliers, and gender. So we look at Thalamo-Cerebellar DMN connectivity as an example here. So assuming all other variables remain equal. So for example, if you have 2 participants that are both 23-year-old males with 30 scan outliers. So if the first participant has a duration of illness of only 1 year, so data was collected at time point 1, then their Thalamo-Cerebellar DMN connectivity is predicted to be 0.0162. Whereas the second participant with similar characteristics, so also a 23-year-old male with 30 scan outliers, but has a longer duration of illness, so the data was collected at time point 4, then the Thalamo-Cerebellar DMN connectivity is predicted to be negative 0.024. So what we have shown is that thalamic connectivity is not uniform across the early stage of psychosis, and it's important to take duration of illness into account, as results can be very different depending on which part of the trajectory participants are at when data is collected. So to conclude, compared to controls, patients have higher thalamic functional connectivity with the visual, motor, dorsal attention, and limbic networks, and lower functional connectivity with the cerebellar, DMN, and FPN networks. Different networks also have different trajectories. Some networks are relatively stable over time. Some networks follow a more curvilinear trajectory, such as the cortical, DMN, and FPN, and some networks follow a linear trajectory. Finally, time since age of onset is indeed a significant predictor of thalamic connectivity within the early stage of psychosis, and this is a potential source of heterogeneity that is not taken into account if the time dimension is collapsed. So what this means is that patients may not necessarily be a homogeneous population because it depends on which network in particular we're studying. So this study has some limitations. So the first is that the thalamus ROI that we used was a whole thalamus. The second was that we had uneven sample sizes at the different time points, especially at the later time points, which had smaller sample sizes. And lastly, because some of our data, our data set was semi-longitudinal, there were some gaps in the longitudinal data. So for future studies, it will be interesting to see if similar results will be obtained with a functional pastillation of the thalamus. It will also, of course, be more ideal to have comprehensive longitudinal studies with even sample sizes and even gaps in time between participants. And finally, given that thalamic connectivity has also been shown to be abnormal in clinical high-risk populations, it would be interesting to recruit subjects at the prodromal stage and then follow them through the early stage of psychosis. So this work could not have been done without the support of my PI and members of the P&L lab, as well as with the help from collaborators at McLean Hospital and the Harvard Medical School. We would also like to thank Professor Fitzmaurice for help with the statistical modeling and the NIMH for funding this research. So thank you.